Hey there, science enthusiasts! Today, I've got some exciting news from the world of cancer research. Researchers at the Francis Crick Institute have just mapped all the possible outcomes of changes to a crucial tumor-suppressing gene called VHL. This breakthrough is the first step in a massive research endeavor to understand the outcomes of tens of thousands of genetic variations linked to cancer. So, what does this mean? The VHL gene plays a vital role in our body by suppressing tumor growth. However, genetic changes or variants can alter the DNA's building blocks. Both acquired and inherited variants in the VHL gene can increase the risk of kidney cancer. Unfortunately, many people are diagnosed with variants of unknown significance, leaving them clueless about their cancer risk. In a study published today in Nature Genetics, researchers used a groundbreaking method called saturation genome editing to track over 2,000 different VHL variants in human cells. They measured cell survival with each variant and gave it a function score. The lower the score, the more harmful the variant. Most variants didn't impact cell survival, suggesting that people with these variants may not have a significantly higher risk. However, some variants were flagged as harmful for the first time, meaning patients with these variants might now receive regular screenings to reduce their cancer risk. The most harmful variants caused cell death, and researchers discovered that the faulty VHL gene increased the activity of another gene called HIF. While HIF helps cells survive in low oxygen, too much HIF can cause tumors. Interestingly, removing HIF from cells with VHL mutations kept the cells alive. This finding shows that the negative effects of VHL depend on HIF. Here's where it gets even more fascinating. A drug called belzutifan inhibits the HIF protein and only works for people with mutations that impact HIF levels in cells. The scoring system developed by the Crick team could identify those who would benefit from this drug. Researchers compared their scoring system with publicly available kidney cancer databases and found that their method could predict cancer-causing variants with 100% accuracy. This means patients with the lowest scoring VHL variants tend to get kidney cancer at the highest rate. Greg Finley, group leader of the Genome Function Laboratory at the Crick, believes this function score could become a clinically useful test to categorize patients. The lab is now working to apply this method to more genes, aiming to bring this level of diagnostic precision to more people. The study involved collaboration with Zhenya Ivakin's lab in Toronto, the Crick's Cancer Dynamics Laboratory, and other institutes in the UK and Germany. This research was funded by the Francis Crick Institute, Cancer Research UK, and other organizations. So, what's next? The team is partnering with the Cancer Dynamics Laboratory to track how well the function score can predict tumor growth and treatment response in patients with VHL mutations. They're also working with the Wellcome Sanger Institute and the Institute of Cancer Research London to map all variant effects across 15 other cancer risk genes. This breakthrough brings hope to countless people living in limbo about their cancer risk. Stay tuned for more updates in the fascinating world of genetic research. Thanks for watching, and don't forget to like, subscribe, and hit the bell for more science insights. Remember, the video is designed to form a deep connection with the audience. The goal is to make complex scientific research accessible and engaging to science enthusiasts.